Hello, my name is Jessica Zehnpfennig, and I'm a PhD student at Central Michigan University. Today I'll be talking about the most recent work I've been doing in the Mahon lab, utilizing whole mitochondrial genomes to resolve evolutionary patterns and internal relationships within Pycnogonida, a class within Trna. To date, there are 10 putative families and over 1,300 described species within Pycnogonida. Pycnogonids are commonly referred to as sea spiders due to their uncanny resemblance to arachnids found on land. However, significant advances in the field of invertebrate phylogenomics has resolved Pycnogonida as a sister to the other chelicerates, with sea spiders being more closely related to Xiphoshira or horseshoe crabs than arachnids. Interrelationships in higher-level phylogeny within Pycnogonida remains highly debated and unresolved. This can be attributed to multiple factors, the first being that sea spiders have high morphological plasticity, with exceptions found for every morphological character state within families and genera. Secondly, the phylogenetic classifications based on morphology is difficult due to high-level phenotypic plasticity within pycnogonids. Lastly, rare, cryptic, or poorly sampled pycnogonid families such as Ostrodecidae and Rhynchothoracidae have been omitted in some previous works, thus there is little known information to help resolve their evolutionary relationships. Mitochondrial genomes are commonly used as molecular markers in the field of animal phylogenetics and have been used to resolve relationships within multiple groups of invertebrates. Proven to be relatively easy to sequence, assemble, and annotate, mitochondrial genomes typically have a moderate level of gene order conservation among related taxa, and changes in gene order arrangements can be used to draw phylogenetic inferences. The goals of our study were to investigate differences in mitochondrial gene order conservation and investigate familial-level pycnogonid evolution and interrelationships utilizing whole mitochondrial genomes from representatives of all 10 putative families within Pycnogonida. The specimens used in this study were collected via Blake trawls during multiple research expeditions aboard the RVIV Nathaniel B. Palmer and ASRV Lawrence M. Gould. 3 of our samples were also collected from sources outside of our group's research cruises in Antarctica. Our second Indiidae sample was collected via scuba thanks to the Moran Lab at the University of Hawaii, Manoa. Our Tanastylum sample was collected via scuba in Belize via Dave Branson, and 1 Foxyclididae sample was provided by Jamie Maxwell at NUI Galway. Additionally, 6 previously sequenced complete pycnogonid mitochondrial genomes were added to the study from NCBI. Genomic DNA was extracted from the pycnogonid muscle tissue with the QIAGEN DNeasy blood and tissue kit according to manufacturer's recommendations. Samples were then submitted to commercial sequencing facilities for library preparation and sequencing. The DNA extractions were sequenced on an Illumina HiSeq 4000 platform with a 2x150 paired N v2 chemistry. The resulting paired end sequences were then checked for quality using FastQC and assembled with Novoplasty version 4.3.1 with the previously published whole mitochondrial genome of Achillea tuberculata as a reference sequence and the CO1 gene as a seed file for the program. Novoplasty was unable to output the mitochondrial genome of Ostrodecidae, so the paired end sequences for that sample were then assembled using SPADES version 3.13.0 with default parameters. The resulting contigs were screened with BLAST-N to determine if they hit with the highest percent query cover a previously published pycnogonid mitochondrial genome as a baseline for mitochondrial DNA identification. The contig containing the mitochondrial genome for each sample was annotated with the Mitos web server and checked manually in Artemis 17.0. We were able to produce whole mitochondrial genomes from 1 Amethyidae, 1 Ascaringidae, 1 Ostrodecidae, 2 Calopelinidae, 3 Colossindeidae, 2 Rhynchothoracidae, 2 Indiidae, 2 Nymphonidae, 2 Palinopsidae, 1 Foxyclididae, and 2 Pycnogonidae species. The resulting phylogenetic tree was created using the protein coding gene data from all of the pycnogonid mitochondrial genomes in the study and the mitochondrial genome belonging to Limnos polyphemus, the horseshoe crab, as an outgroup. We used IQ-Tree2 to construct the phylogenetic tree using a partitioned maximum likelihood analysis and the best fitting model for each gene. This resulted in a consensus phylogeny of sea spider families. We assessed the strengths of the resulting relationships based on 1,000 rapid bootstrap replicates. All pycnogonid mitochondrial genomes sequenced and annotated in this study were complete, containing 13 protein coding genes, 2 ribosomal RNA genes, and 22 tRNA sequences, along with an AT-rich control region often seen in arthropod mitochondrial genomes. The assembled mitochondrial genomes ranged in size from 14,650 base pair in Calopelinidae to 17,834 base pairs in Foxyclididae, averaging 15,032 base pairs. All Pycnogen mitochondrial genomes were found to have consistent protein-coding gene order and directionality or strand placement, with no variations found among the different families, with the order and directionality of the protein-coding genes the same as previously published chilosert mitochondrial genomes. Within our dataset, Amethyidae, Ascarinchidae, Ostrodescidae, Classendiidae, Palinopsidae, Rhynchothoracidae, and indeed species number 2 all have the same tRNA arrangement, which differs from the accepted ancestral arthropod tRNA order found in Limulus polyphemus, the horseshoe crab, in placement of tRNQ with respect to tRNI. India species number 1 showed a different rearrangement in tRNQ and G as well as tRNN. Foxycholidae also showed a rearrangement in the placement of tRNC. Representatives of Caloplinidae and Nymphonidae show rearrangements in tRNA, E, Y, P, S1, V, Q, M, and I that differed from other arrangements found in other families or other chelicerates. Past research has shown that there are 2 common regions in Trna translocations within arthropods, one near the large non-coding control region and the second near the region that contains tRNA, R, N, S, E, and F. We see this in Nymphonidae and Caloplinidae. These translocations can indicate 2 things. One, that there is a potential for a second strand origin in Nymphonidae and Caloplinidae mitochondrial DNA near that region, or 2, due to the lack of introns in both families, the Pycnogen mitochondrial order exhibited in Nymphonidae and Caloplinidae is ancestral to Pycnogonids. The other Pycnogonid families had translocations in tRNA genes near the long, non-coding introgenic regions, which could be evidence for the duplication random loss model for mitochondrial DNA rearrangement. This is where partial duplications of mitochondrial DNA occur due to slip strand mispairings, followed by the loss of one copy of each duplicated gene. This results in an intermediate state or pseudo-sequence remnant within the duplicated region. The presence of intergenic, non-coding sequences near the translocated tRNA sequences found in our data suggests that these introns may be pseudo-sequences or remnants of past duplication. The control regions of all Pycnogen mitochondrial genomes were found to be a large AT-rich repeat region located near the 12S rRNA gene, with placement varying slightly by family, often occurring between tRNQ and tRNI. tRNQ is considered a translocation from the accepted arthropod mitochondrial DNA order, thus the control region may also play a role in gene order translocation throughout Pycnogonida. Multiple non-coding AT-rich introns, over 100 base pairs in length, were found located within multiple Pycnogen mitochondrial genomes in our study. This is commonly seen in arthropod mitochondrial DNA, with small intergenic regions having been reported within Ligia oceanica, as well as large non-coding regions with multiple repeats being reported within species of bark weevils and marine annelids. One explanation for these introns could be the possibility of Pycnogonids having multiple control regions present in their mitochondrial genome, as seen in the mitochondrial genomes of phytoseed mites. Another plausible explanation is that the large introns may be a derivative from ancient transposable elements and have lost any potential function. These introns could also be attributed to tandem duplication random loss events, and they could be an indicator of evolutionary rates in multiple Pycnogonid families. Non-coding mitochondrial sequences are known to be fast evolving due to strong mutation pressures and the shared placement in some of these introns, as seen in Amethyidae and Classendiidae, which could indicate the presence of these introns in the common ancestors of families. Interestingly, the CO1 gene of our Tanastylum species in our study displayed a group 2 intron of 997 base pairs within the gene. This intron may have occurred as a discontinuous part of a transposable element that is split among the gene and transpliced to form a functional intron within the gene. The intron could also be a result of recent horizontal gene transfer from a bacterial or viral intermediate, as seen and suggested in the mitochondrial genome of the unidentified Nephitis species. As this intron is only present in one of the specimens in our study, further mitochondrial genomes will need to be sequenced to determine the true reasoning behind its appearance. The Phylogenetic Tree recovered 6 clades within Pycnogonida. A clade consisting of Nymphonidae and Caloplinidae were placed as the sister clade to all other Pycnogonid families. Classendiidae and Pycnogonidae formed one monophyletic clade, and Indiidae and Foxycholidae formed another within Pycnogonida. The 4th clade recovered placed Palinopsidae as a sister to the remaining taxa, and the 5th clade recovered contained Rhynchothoracidae as a sister to some members within Amethyidae, including both Tanastylum species and Acheliobit tuberculata. This clade was placed as a sister clade to the sixth clade that contained Ascarinchidae, Amethyidae, and Ostrodecidae. Within this clade, Ascarinchidae was placed as a sister to Amethyidae and Ostrodecidae, and Amethia carolinensis was placed as a sister branch to the monophyletic clade containing Amethia calmani and Ostrodessus species as sister branches. The phylogeny produced by our data is a novel familial level phylogeny that contrasts the results of previous studies utilizing molecular and morphological data. The plasticity within Pycnogonida negates the ability to rely on morphology for deep level relationships, and the findings of this study highlight the importance of the use of molecular data in constructing sea spider phylogenies. Examining the full mitochondrial DNA showed variation in different Pycnogonid mitochondrial genomes. We found that while protein-coding gene order is consistent through all of the families, there is a variety of tRNA translocations between families as well as species. Mitochondrial genomes are considered fast-evolving when compared to nuclear genomes, and thus could exhibit limited utility in resolving relationships of sea spiders without the additional molecular data. Our data also created a novel level phylogeny for Pycnogonida families. It is important to note that the use of mitochondrial genomes in phylogenetics can be limited due to the high rate of evolution in mitochondrial DNA, which could result in incomplete lineage sorting and lead to long branch attraction artifacts. Our study contained a limited sample size with 1 to 2 specimen used per family. The addition of different species may give more insight into interfamilial gene order and conservation, as well as interfamilial phylogenetic relationships within Pycnogonida. And with that, I thank you for your time, and I would love to take any questions.